Hey everybody, welcome. My name is Amy Trowbridge and I'm an assistant professor in the entomology department here at UW. And it is really my pleasure today to talk to you a little bit about the field of chemical ecology. What I'm going to do in this first video is just give you a brief history of chemical ecology and some of the different compounds that you might be familiar with that mediate insect and plant interactions. And then in the second video, I'll talk more about those specific compounds and some really cool ways that they affect herbivores. And again, try and give some examples of things compounds that you might be familiar with. So what I'm going to do right now, though, is minimize my, I just wanted to say hi. I'm going to minimize my picture here so that I don't have to move my face around too much and I can just talk to you and you can focus on my slides and maybe even enjoy some soothing talk. Okay, so let's get started. If I can advance my slide, there we go. Okay, so one of the first questions you might be asking to yourself right now is, what is she talking about when she says chemical ecology? And maybe also, why would I care about chemical ecology? Well, since you're in this class, I'm assuming that you might be interested in chemical ecology because this is a field that can really reveal processes that drive kind of that variation in ecosystems, that phytochemical variation in ecosystems, and then helps us understand how that chemical variation mediates interactions with all of the different organisms in that ecosystems, but in particular, insects. And so really what chemical ecology allows for is this mechanistic approach to understanding the causes and consequences of species interactions, species distributions, their abundance, but also their diversity. And so more specifically, this field really aims to understand the role that chemistry plays in mediating a lot of different types of interactions. And so one you might be familiar with is mutualism, right? And so this is a bee and a flower, and we're talking about pollination. And, you know, the chemical ecology of pollination is fascinating because there are all these chemicals that are involved in color and how color is determined in flowers, but also in their scent. So that those olfactory cues, and also the taste of the nectar and pollen, which are these gustatory cues and rewards. So chemistry plays a really big role in those interactions, which I wish we could spend an entire course talking about. Another one would be parasitism. So where parasitoids, for example, they can find their herbivore hosts because the herbivore feeds on a plant and releases very specific volatiles that the parasitoid has learned to associate with their host. And then they come and lay their eggs inside of this caterpillar, and the eggs then hatch. The larvae eat the caterpillar from the inside out and then pupate on the outside. Again, another topic I wish we had at least a semester to talk about together. Also, chemistry mediates predator-prey cycles. So this is, Daphnia and a tadpole, and they can actually change their development based on whether or not they perceive chemical secretions from their predators, which is just so cool. And then finally, just this idea of resistance and defense. So I'm showing you here a monarch. These insects are capable of ingesting and sequestering or taking up cardenolides from milkweed, which serves as protection against predation. This is monkshood. Monkshood produces these really potent neurotoxins that are just obviously used in defense, but are pretty rad when you think about it. So anyway, there are just lots of examples of chemistry mediating all of these incredible interactions. And so I'm going to give you just a small little sliver and taste of that right now in the hopes that it'll pique your curiosity and you'll do some more digging. Okay. So because the topics that are embedded in the modules for both this week and next week focus on how chemistry impacts a plant's ability to resist herbivores, mostly insects, what I wanted to do here is just quickly kind of outline the major mechanisms by which this occurs, right? So first, chemicals within plants can have direct detrimental effects on herbivores, and they do this by mostly serving as toxins And this is in a very dose-dependent manner. And then also they can affect the insect's ability to digest and absorb nutrients from those plant tissues. So these chemicals then, after the herbivore feeds, they become induced, which means they're produced either they're brand new compounds that are produced, or they're just compounds that were there but are produced in much higher quantities. So they're induced in response to that feeding. And not only can they serve as toxins and have these direct effects, but they can also serve as cues for rewards or information for the natural enemies of herbivores, like predators and parasitoids that I mentioned earlier. So a good example of this is through the herbivore-induced production of volatiles, like I mentioned before. Those volatiles that the plant produces when it's being fed upon by different species of herbivores are very specific. And those specific volatiles, or bouquets, if you will, are subsequently just exploited, right, by these parasitoids that through associative learning, kind of like Pavlov and the dog and the ringing the bell with food, it's the same principle here. They locate their host, they lay their eggs, and then oftentimes that eventually kills the herbivore. So this is, of course, great for the plant, right? Because it provides them this form of indirect resistance against continued feeding, which, I mean, what plant wouldn't love that? So, yes. So now I want to talk about, these different types of chemicals that are involved in plant resistance and defense. And we usually refer to them broadly as plant secondary metabolites, or PSMs. But again, they have really like a ton of functions. So we always say defense, but I just want to slip that in here because they do play other roles. And I might touch on that a little bit later. Okay, anyway, the reason that we call them secondary metabolites is because they're not actually directly involved in growth, development, or reproduction like primary metabolites would be. So primary metabolites we consider broadly as like carbohydrates and lipids and proteins. So these aren't those, but even though they don't have a direct involvement in growth, development, or reproduction, they are required for survival. Right. And so another thing that I want to emphasize here is that these compounds, these secondary compounds, actually rely on primary metabolites, those photosynthates and other compounds from cell component synthesis. They rely on those as the building blocks to actually come together to make the secondary metabolites. And so even though they're two separate groups, they are intimately linked. So what are some examples of secondary metabolites? I would bet that you know quite a few just from your own lives and experiences, but I'm going to give you some examples just because why put you through that torture of thinking of them on your own. Okay, so here are some examples that if we were together in person and we could have a discussion, I thought you might come up with. And I'm going to talk about them in the context of the three main categories that these compounds are categorized into. And so I'll go into that in the next video. So don't worry about the three categories right now. You don't have to memorize them, but just to give you some context. So the first category are called nitrogen containing compounds, which obviously these are just molecules that have a nitrogen atom, at least one in that molecule. And so the example I'm going to give here is here in the upper left-hand corner. And some of you might know this plant. I sure didn't until I went to graduate school, but this, my friends, is Atropa belladonna, or deadly nightshade. Now thinking about it, I wish I would have known, because what if I wanted to eat it? No, I wouldn't. Okay, who knows? Anyway, this species, or deadly nightshade, produces a group of compounds known as tropane alkaloids. So what's really interesting about this plant is it has a long history of use. So one example is before the Middle Ages, it was actually used as an anesthetic for surgery. And then another example is that actually the ancient Romans used it as poison, which probably doesn't surprise many of you because they were using everything as poison back then. But anyway, again, it's Atropa belladonna is the scientific name, and I think it's just so cool. Atropa actually comes from the word atropos, which, if any of you are mythology buffs, is one of the three fates. in Greek mythology. And the name belladonna is derived from Italian, which means pretty woman. And the reason that it's called Atropa belladonna is because this herb was used in eye drops to dilate women's pupils, and the Italian women thought that that made them appear more seductive. And so what, maybe you're making this connection, but really where we get atropine from is from this plant, from this compound, which your eye doctor uses to dilate your eyes to do your eye exam. And so I don't know about you, but I look like an absolute cartoon character when I come out of the eye doctor, and I don't feel like a pretty woman, but I don't know. I think maybe the Italian women knew something I don't. And then another nitrogen containing compound, and you can see in this lower right hand, is this is the plant that produces it, which is caffeine, right? So coffee. So caffeine is another secondary metabolite that also affects herbivores, but obviously also stimulates and affects us as well. Okay, so that's nitrogen containing compounds and some cool examples. I'm going to move on to the terpene group. And I thought maybe some of you would come up with a compound made from this plant right here in the upper right. And you might recognize this as cannabis. And cannabis is known for making a lot of THC, or tetrahydrocannabinol. And so, like most pharmacologically active metabolites in plants, THC is assumed to be involved in defense against herbivores. But what's kind of cool is it also possesses this high UVB absorption property, which people have speculated could protect the plant from harmful UV radiation exposure, kind of like a natural sunscreen. Okay, so we've talked about nitrogen containing compounds. We talked about some terpenes, and now we're going to talk about phenolics, which is the third group. So what I want to talk about with phenolics is a group of compounds known as tannins. So these are called tannins because they've traditionally been used to tan animal hides into leather. And the reason that they can do this is because they have a property that allows them to bind proteins. And so you can find tannins in lots and lots of different plants, but you might recognize them in walnuts. And if you have noticed, like me, there are so many walnuts on the ground right now. But you can also find tannins in oaks. And I'm giving you this example of oak barrels here on the bottom. And so the reason that winemakers actually age their wine in oak barrels is because the tannins seep into the wine as it ages. And that's what gives wine those, like, that kind of dry, puckering feel in your mouth. So, okay, those are some examples that maybe you knew about, maybe you didn't, but guess what? We all know now. So, so that's pretty fun. Okay, so I just want to kind of give a real quick overview on just the field of chemical ecology, because it is a relatively new field. It was kind of founded about 50 years ago, and it was kind of a field that came out of this notion that all organisms use chemical compounds to mediate interactions at lots of different levels, right? So this idea that chemical communication is a universal feature of life. It occurs at all levels of biological organization. It helps regulate cells and organs within the body and plants, and it also helps mediate interactions at a larger scale, at the ecosystem level, organismal level, et cetera. And so I just want to talk really quickly about kind of the three major strands in the development of this field. And I'm going to not spend a lot of time here, but just to kind of give you a sense of how did this field even come to be? Well, it came to be because people were noticing things about plants and insects and chemistry. And one thing they noticed is, wow, there's a lot of secondary metabolites in plants. One question we have is, why the heck are they here? Another thing they noticed is that very small amounts of very simple compounds had really profound effects on insects. And the third thing was that insects that could take up these compounds or sequester them tended to have kind of this warning coloration or aposematic coloration. And so all these questions kind of led to the development of this field. And I just want to talk really briefly about all three, but I'm going to start here with this need to explain variation in secondary metabolites in plants. And so I just want to start here by saying, first of all, that plant herbivore interactions go way back. The types of herbivore damage that we recognize today were apparent millions of years ago. And you can see that in this upper right-hand corner here. This is evidence here from 55 million years ago of herbivores chewing and mining and creating galls. And so obviously insects have been interested in plants for a very long time, but so have humans, right? Our human interest in natural products also goes way back, but obviously not 55 million years. So this picture on the left here is from a 15th century manuscript illustration, and it's showing you a mandrake root. Actually, it's very Harry Potter-esque if you've ever watched that movie or read the books. But anyway, throughout history, mandrakes have really they were really highly sought after because they have aphrodisiac properties. But the roots of the mandrake plant also contain large quantities of alkaloids. Scopolamine is one of the main alkaloids in the mandrake root, and at high doses, it can affect the central nervous system and act as a depressant. And so it makes the plant actually really toxic to herbivores. But kind of interestingly, like from a human perspective, scopolamine was actually used later for pain management and in labor specifically. I don't know if they use it anymore, but I do know that in small doses, it's still used today to prevent motion sickness, which is kind of cool. But anyway, noticing all these things, people were asking, well, what is the function of these compounds? Why would a plant make these compounds? And you can see here in the lower right that Gottfried Frankl gave one possible explanation, which was defense. And not only that, but insects that could evolve and adapt to those compounds might actually not be affected by them in terms of defense, but might actually use them as attractants. And so he says here, these odd chemicals arose as a means of protecting plants from insects and now guide insects to food. And that leads me to this idea of coevolution, which I'm just putting the slide here as a teaser because it's such a cool topic, and we'll dig into that more next week. But when you think about specialists and how they've evolved, you have to discuss coevolution. And just, I love this quote here about how it takes all the running you can do to keep in the same place, where plants and insects are constantly evolving to keep up with one another. And this is known as the Red Queen hypothesis. And we'll talk about that more in a bit. Really quickly, I just want to say something about trace compounds and how they can have profound effects. And I'm not going to focus on chiromones here, but I just want to talk mostly about sex pheromones in the order Lepidoptera, so with moths especially. And basically, it was observed that 1 microgram or less of simple organic molecules could actually have profound effects on the mating behavior of moths. And I'm specifically saying moths, and this is a moth because of these beautiful feathery antennae. But I'm talking about moths specifically because moths are diurnal and they don't have very good eyesight. So they need to find each other for mating using pheromones. Whereas butterflies, they fly during the day and they have much better vision. But anyway, it's just so incredible. I mean, there's a fact that the male emperor moth is able to respond to a female releasing pheromones from several miles away. I mean, that is bananas. But usually, more typically, pheromones are effective over about 100 meters or so. Okay, so then finally, I just want to talk about aposematic coloration and sequestration. So, insects can have these beautiful distinctive markings or colors that obviously contrast with their background, and it serves as a warning to predators not to mess with them. And so most species that have this type of coloration, besides mimics, are capable of taking in these toxic compounds from their plants' food source in the larval stage. They can store them in their integument, and then they can move them into the adult phase so that they can be protected from predation. And this is just such a classic, cool example of a blue jay feeding on a monarch and then instantly regretting its decision and puking the monarch up. And the reason is because monarchs can actually sequester the cardiac glycosides that are present in milkweed, which is its host plant. Okay, so obviously a lot of what drove the development of this field of chemical ecology was this observation at different levels that secondary metabolites play a major role in plant defenses against insects. And so, what I just want to stop here and kind of just let you know that the next time you watch a video from me, you will learn more about the defensive roles of these different compounds. And I'll give some more examples of just kind of, the properties of these compounds that allow them to be used as defenses against insects. And it's very cool and fascinating. And yeah. Okay, so I'm going to stop there, and thank you so much for listening. And I'm looking forward to having you participate in listening to the next video. So thanks, everyone.